Hello Mebane. Welcome to the Water Resource Recovery Facility. I am John Dotson, the director here of the operations. Today what we're going to be looking at is, we'll be in the lab looking at a couple of things. We'll be looking at some of the process control testing we do, and also, kind of really neat, is some of the microorganisms that actually do a lot of the work out here to produce the clean water that leaves our facility. All right, well, let's go check out and see what we can see.
My name is Amanda Hill. I'm the laboratory supervisor for the city of Mebane. And today we are going to look at how the treatment process works, how we test the treatment process to make sure it's working. So this is raw wastewater, and this is what it looks like when it comes in the plant. And at the end of 24 hours, which is how long the treatment process takes, we have a nice clean effluent to return to the creek. Well, today we're going to look at everything that happens in between so that we can make sure that the process is working correctly. So the treatment process is actually a biological process. And so all of this brown stuff you see at the bottom here is our bacteria that actually treats the wastewater for us. So all of the pollutants that come in the raw wastewater is actually food for this bacteria. So one of the tests that we can run is to make sure that the bacteria has a nice home, that it can flock together. And so this is a very simple physical test. It's called a 𝒖𝒓. And so we'll mix all of this bacteria up with the water and then simply see how well and how fast the bacteria can settle from the bottom and separate from the water that it has already cleaned. So here we have a real-time view of what we see under the microscope. This right here is a rotifer, and so this is one of our protozoans. And even though we're not seeing the bacteria that does the treatment for us, what we're seeing is we're seeing the predatory protozoans and metazoans, the other creatures that actually eat the bacteria. So if we find these creatures under the microscope, we know that there's enough bacteria as food for them, so we know we have a good bacterial load. Up here you'll see, kind of running around, is a free-swimming ciliate. Move that around. And so that's another one of our critters that we look for under the microscope. And you'll see that they're crawling around on what we call this flock, and that is actual bacterial secretions that create homes for the bacteria to live in. And when you see this nice flock where it's all sticking together good, we know it's going to settle good in the 𝒖𝒓 because they flock together and become more dense than the water, so they can actually settle out. So we're also looking for that. Hey John, can you find me a filament?
Yeah.
So this right here that you can see moving around, that's a nematode. It's a type of worm. So that's one of our higher order organisms, so that we can tell when we see something like that what the age of our bacteria is. And so we can see it's a little bit older of a bacterial load, which is what we actually want in the type of system that we have. And then if you see what kind of looks like tree branches coming through this flock, those are actually filamentous bacteria. Those are very large bacteria, so we can actually see those under the microscope. But it's nice to see some of those because they act as a scaffolding for the flock to be able to hold it together. So another thing that we're looking for under the microscope is, if you can see this piece of organic material right here, that is actually a piece of 𝒂𝒓, and that is the organic material that we actually add to the system to create a place for the flock to attach to. So we like to see all of that flock attached to the 𝒂𝒓 because, again, we know it will settle really nicely. So this is actually another kind of rotifer. So we have several species of the same type of creature in here, and seeing that biodiversity is something that we look for as well. Here's a pair of rotifers. You can see both of them feeding at the same time, and their little mouth parts are actually open with the ciliated edges, and they're pulling bacteria into their mouth parts. And then down here, what looks like a heart, that's not actually a heart, that's their mouth, their internal mandibles, where they're actually eating their food. So one of the pollutants that we want to make sure is being removed during the treatment process is ammonia. And in order to remove ammonia, the bacteria has to have plenty of air available to it. The bacteria is aerobic, just like us. It needs to breathe oxygen. So in order to check that, we can run a quick ammonia test. And as long as the bacteria has enough oxygen, there shouldn't be any ammonia in this process. So here we just add a couple of drops of our reagent to one of the tubes so we can compare that there is no ammonia during the treatment process. So we know we've added enough air for the bacteria to be able to remove the ammonia from the water. So ammonia is only one form of nitrogen we want to remove. Nitrogen is a nutrient that we don't want to put too much out into the environment because it causes algae to grow. So another form that we want to remove is nitrate, so NO3. So we test for nitrate as well during the process. And we do that using a spectrophotometer. And all this does, it's another colorimetric method where the nitrate reacts with the reagent and turns a certain color. And then we pass a beam of light through that sample, and some of that color or some of that light is absorbed by the color, and whatever passes out on the other side is measured, and then we can see how much nitrate is in there. But we don't have to do that by hand anymore because spectrophotometers are automated. So one more test we'll do on the treatment process is testing for phosphorus. That is our other nutrient that we're concerned about being in surface water, because phosphorus is the limiting nutrient in fresh water. So we want to make sure that we're getting rid of phosphorus during the treatment process before it reaches the creek. So here we'll add a reagent that turns blue in the presence of phosphorus. And this first vial is just deionized water because we want to know what 0 looks like to be able to tell the machine what 0 is. And then we'll add our reagent to the actual sample. Mix it up. And we'll wait 2 minutes for the color to develop. All right, so because hopefully we have removed all the phosphorus during the treatment process, we will also run a standard, and this is where we add a known amount of phosphorus to a sample so that we can actually see what the sample would look like if there were phosphorus in it. All right, so after 2 minutes, we've had enough time for the color to develop. And so just like the nitrate, we read this on a spectrophotometer, pass light through the sample, and measure what comes out on the other end. The more that's absorbed, the higher amount of phosphorus that will be in the sample. So the first thing we're gonna do is we're gonna take our deionized water that we tested, that we added reagent to. We're gonna tell the machine what 0 is. And then we're gonna take our standard that we ran. This is a known standard, so this should be around 0.5 milligrams per liter of phosphorus. And we'll read that. We got 0.51, so perfect. So at 0.51, we know that the test is working correctly. And so now we can actually test from the aeration basin, our sample that we put in here, and see if we have any phosphorus in the system. So our process sample read 0.04 milligrams per liter, and actually this particular test has a lower detection limit of 0.1 milligrams per liter, so we just call this a less than. So we have removed phosphorus in the process.
So here's a rock out of the effluent. We have a 𝒖𝒓 𝒂𝒂𝒏𝒊𝒔𝒎𝒔. We have some more caddisflies. This is another type of caddisfly. We have some small worms. We have snail eggs. The caddisflies are part of what's called the EPT, which is the Ephemeroptera, the Plectoptera, and the Trichoptera, which are 3 orders of aquatic insects that are highly pollution intolerant. They cannot live in polluted water, especially the green caddisfly, which is a 𝒖𝒓 𝒂𝒂𝒏𝒊𝒔𝒎𝒔. It means water spirit. And they are completely pollution intolerant. So if the water was in any way negatively impacted, those organisms could not live here.
We hope you found this video informational and helpful. And I'll end with this. There's nothing positively charming about poo. However, when you have a team of dedicated professionals that can take wastewater and turn it into something that's a valuable resource to the community and the environment, that is positively charming. Again, we hope you found this video helpful and informational, and thank you for your time.